Hello everyone. So today I would like to start this podcast with few stats and numbers. Every minute, approximately 250 children are born somewhere around the world. 56 of them were relying on seafood as the primary protein source. Now this means that every year, approximately 530,000 new people were relying on seafood. Our oceans simply can't sustain these rhythms. And it is obvious that if we keep fishing the way that we are, then very soon we will not have much seafood left. So we currently stand in front of a sustainability scale. On one side, we have a fast, hungry, growing population, and in the other, the welfare and future of our oceans. I'm Dr. Roberta Markley, and I'm an aquaculture researcher and science communicator. I strongly believe that the answer to this is good, sustainable aquaculture production. I've committed my career to applying molecular biology and genetics to find practical solutions for the aquaculture farming so we can boost the welfare and future of our ocean while feeding the world with nutritious seafood proteins. Aquaculture is the largest growing sector globally as it can produce sustainable proteins and help feeding the planet. However, unlikely terrestrial farming, there is not centuries of research into finding the best condition for farming those animals. For example, the implementation of breeding programs in the aquaculture sector is not being used extensively yet, and the understanding of the physiology of aquatic animals still has a lot of ground to be covered. The implementation of cutting-edge technology such as transcriptomics, epigenetics, and proteomics can help us better identify traits of interest, but also prevent, predict, and help with the optimization of farming conditions. I've worked with multiple aquatic species and have implemented the use of molecular biology as a tool to monitor health, predict, and characterize phenotypes. And today I will talk a bit about my latest two papers, one on climate change and one on fish implementation, both published in 2023. So it is widely accepted that climate change is and will be a threat to the aquaculture industry, with the biggest issue being a slow but continuous rise of the water temperature and associated decrease in dissolved oxygen in some areas. New Zealand fish farms during the hottest period of the year, which is January, February, the increased water temperature has been shown to be detrimental to Chinook salmon performance, and increased mortality have been observed. With the study I'm here presenting, we have monitored the response of Chinook salmon, that by the way is also called king salmon, to chronic long-term increase in temperature and decrease in oxygen over a 3-month period. We have also identified, characterized, and developed biomarkers for Chinook salmon and applied them to monitor responses to changes in fish environment. So through this study, we have demonstrated that even a small change in temperature, so from 17 to 19, can affect the animal drastically, resulting in a 45% mortality at the higher temperatures. Through these discoveries and the other associated one, the industry has now decided to move their farms offshore in open ocean farms to resolve for now the effects of climate change on this important industry. Another great example of the implementation of multiomics and multidisciplinary approaches in the aquaculture sectors is the understanding of the grey phenomenon in barramundi. Barramundi, also called Asian sea bass or Latus calcarifer, is one of the most iconic fish in Australia. Loved to go out and catch, but very profitable and important for the aquaculture industry. However, barramundi often exhibits graying in the flesh, a normal and physiological appearance that, however, is not appreciated by the customers. In fact, a study done in 2017 has demonstrated that fillet coloration is one of the most important factors when purchasing fillets. Here in Australia, the barramundi industry is predicted to expand exponentially, but it is obvious that with the willingness to expand, customer choice becomes a very important factor. So in collaboration with our industry partner, Mainstream, here at James Cook University in Townsville, where I'm currently working, we have been exploring the reason behind the graying phenomena and possible approaches to reduce it. Interestingly, we have found a very rare phenotype, golden barramundi, where the skin of the barramundi is completely golden, but as well as that, these animals have perfect clear fillets, not showcasing the graying issues. So we thought that if in one hand we can produce more of these golden barramundi, and in the other we can use the golden phenotype as a model of understanding the grain phenomena, we could help the industry to reach their goals and reduce this grain issue. Therefore, through implementation of multiomics approaches, we have found that the reason behind the graying in the fillet is correlated with melanin, and the golden phenotype has a reduced quantity of melanin-producing cells that are also called melanophores. The golden phenotype also has several downregulated genes related to the melanin production, and those changes have been correlated through our methylome study to epigenetic signature. We are now trailing the utilization of CRISPR-Cas technologies to target some of the genes related to the melanin biosynthesis as a proof of concept of our study. But as well as that, we are now trying to find the condition that can trigger the golden phenotype. So if we can produce more golden barramundi, or at least reduce the melanin presence in the wild-type fillet, the industry will be able to be more profitable with a fillet that is much more appreciated by the customers. To conclude, these are just two examples of the incredible work that can be done by implementing multiomics approaches in the aquaculture sectors. Through understanding and researching these aquatic animals, we can help the aquaculture industry be more productive, sustainable, and resilient while feeding the planet with nutritious seafood proteins. Now, if you have any question about my work, please reach out either via email or LinkedIn. But thank you for listening.